everyone, thanks for the invitation to speak today. I'm going to talk about our work looking at the protective role of the protein disulfide isomerase family of proteins in a neurodegenerative disease, amyotrophic lateral sclerosis or motor neuron disease, depending on which part of the world you come from. And I'll also describe how some work that Marcello Camini's team have done with a biosensor has really helped us in this study. And first, I'd like to start with the acknowledgements. So most of the work that I'm going to be talking about today was done by Sonam Parak, who's a postdoc in my team. And I just want to thank also the hard work done by Marcello's team, particularly Claudia, Morella, and more recently Cecilia in this project. And if anyone is interested to know more, here are some of the publications that describe this work. So the first paper at the top is from iScience. It was published last year, and this really describes the bulk of the work where we show that the redox activity of PDI is what's mediating its protective role in ALS. And this also describes the studies with the biosensor. This year we published this publication where we've extended these findings to another protein that's linked to ALS, FUS. And at the bottom is a paper that's on bioRxiv where we've identified another mechanism that PDI is protective against, which suggests it seems to be protective in the nucleus. And if anyone's interested in a general review article about redox dysregulation in the pathogenesis of ALS or MND, this is a review article that was also published this year. So ALS is a neurodegenerative disorder affecting motor neurons in the brain, brainstem, and spinal cord. So ALS affects both upper and lower motor neurons. So the upper motor neurons connect the brain to the spinal cord, and lower motor neurons connect the spinal cord to the muscle. So ALS usually starts with a fairly painless muscle weakness, and patients may often notice that they begin to trip over or they start to drop things. But it progresses quite rapidly until there's almost complete paralysis, and it's usually failure of the respiratory muscles that's the fatal event. It's a disease associated with midlife, and death usually occurs within 3 to 5 years of diagnosis. It has a prevalence of about 6 cases per 100,000 individuals, but given the aging population, which is predicted to increase dramatically over the coming decades, the prevalence of ALS is thought to increase significantly. And there's no real effective treatment. So there are 2 possible FDA-approved drugs, but neither are particularly effective, and they only prolong life by a few months in each case. So there's really an urgent need to develop more effective treatments. So most ALS arises sporadically, about 90%, but about 10% of cases are familial, and there's now a long list of genes that are known to be mutated in ALS. I'm not going to talk about the most common mutation, at least in Caucasian populations, which is C9orf72. However, I am going to talk mainly today about the next most common mutations. So these are in superoxide dismutase 1 or SOD1, which is about 20% of familial ALS, and also about 5% of cases are caused by mutations in TDP-43 and FUS. However, ALS is the 2 forms of ALS, sporadic and familial, are clinically and pathologically very similar. Unlike most neurodegenerative disorders, protein misfolding and aggregation is really the key pathological hallmark. So the first of these proteins, superoxide dismutase 1, which is predominantly found in the cytoplasm, and it has an antioxidant function in detoxifying superoxide radicals. So the mutations in ALS are scattered throughout the whole protein pretty much. And what's interesting is that whilst SOD1 itself is a dimer, the disulfide reduced monomer, so it normally has an intra-subunit disulfide bond, this becomes reduced in ALS. And these monomers seem to form non-native disulfide bonds, which are found in the aggregates. TDP-43 is quite a different protein to SOD1. It's normally found in the nucleus, and it binds both DNA and RNA. And it has an increasing number of functions. It's most recently been shown to be a DNA repair protein. So that was work done by our group and several others. However, in ALS, TDP-43 mislocalizes to the cytoplasm. Similar to SOD1, abnormal disulfide bonds seem to be involved in aggregation, and this seems to be induced by oxidative stress. And the mutations are also found throughout the protein, but do tend to cluster in the C-terminus. And TDP-43 is particularly important in sporadic ALS as well, as I'll mention in a minute. FOS is a very similar protein to TDP-43. It's a DNA-RNA binding protein with also functions in RNA metabolism and DNA repair. It's found in the nucleus normally, but it also mislocalizes to the cytoplasm in ALS. However, with FOS, these aberrant disulfide bonds have not yet been described in ALS. Similar to TDP-43 though, these pathological forms are very important in, it's now thought to be in sporadic disease. So these pathological inclusions or aggregates are a sign of protein misfolding, and this is really the key pathological hallmark of ALS. And in the familial forms of ALS, it's these proteins, SOD1, TDP, and 43 and FOS, and other proteins that are mutated that form these inclusions. However, what's interesting is that in sporadic ALS, wild-type forms of the same proteins seem to misfold and form inclusions as well. And TDP-43, it's been long established that TDP pathology is present in almost all ALS patients. A study showed that this was about 97% of all ALS cases. It was originally thought that FOS wasn't implicated in sporadic disease, but recent studies have shown that similar to TDP-43, FOS is mislocalized and forms aggregates in sporadic ALS. There's many different pathogenic mechanisms implicated in ALS, and I haven't got time to talk about all of these today, but I just want to highlight a few and mention that really they seem to cluster into two main areas. Firstly, in DNA and RNA metabolism, which is probably not surprising given that TDP-43 and FOS are involved in this mechanism, but also in proteostasis. So there's many different aspects of proteostasis that are thought to be dysregulated in ALS. I'll talk about a few of these today. Mislocalization of TDP-43 and FOS, as I mentioned, is one particularly important mechanism. ER stress, protein misfolding, and also some forms of cellular transport are also implicated in ALS. So we've been looking at PDI for a while in ALS, and what is PDI? It's an endoplasmic reticulum chaperone. And I say it's conventionally thought of as being an endoplasmic reticulum chaperone, but it's actually found in other locations as well. And an increasing number of incidences of PDI being localized outside of the ER are described. It's particularly well described extracellularly. It contains this structure shown here. So it's got these two catalytic domains, A and A prime, and these domains contain this active site region, CGHC. So it's got these two redox active cysteines in each active site. It also has the substrate binding domains, B and B prime. So it's a general protein chaperone, but it's also important because it's an oxidoreductase. So by this redox function, it actually is responsible for forming disulfide bonds in proteins. So it can act as an oxidase, a reductase, or an isomerase in this process. And PDI is a prototype of an extended family of proteins. There's around 21 members now, and they contain this active site motif, although not everyone, but this is a common motif, this CXXC. I'll mention a few of these family members today, some of the ones that we've looked at. So primarily PDI, but we've also looked at ERP57, which is the closest homologue to PDI, and also ERP72 and PDI-A2. So we first asked whether PDI might be protective in ALS and could be used as a therapeutic target. And this really first came from this idea that PDI is responsible for forming disulfide bonds in proteins. So given that there's this aberrant aggregation by disulfide bonds, could PDI be protective? So we first looked at this about 11 years ago in this study, when we looked at transgenic models of superoxide dismutase 1 or SOD1, and we looked at in cell culture firstly to see whether PDI might inhibit protein misfolding, and we found that indeed it did. So if you look at the blots at the top, this represents insoluble SOD1 and also ubiquitinated proteins, so the aggregates are known to be ubiquitinated in ALS. And when we overexpress PDI, we find that we can reduce the formation of these aggregates. Similarly, when we look at microscopy, and the graph at the bottom shows you this, when we overexpress PDI, we see there's significantly less inclusions formed in cells, and the more PDI we express, the more these inclusions are reduced. Similarly, if we knock down PDI, we see more inclusions. So this was fairly obvious that there was a strong link between protein misfolding of SOD1 and PDI. However, what we also found was that in ALS patients and in a SOD1 transgenic mouse model, that PDI was aberrantly modified by S-nitrosylation. So while, of course, S-nitrosylation can be part of a physiological process, this aberrant modification has been linked to neurodegeneration, which inhibits enzymatic activity. And we found significantly more S-nitrosylated PDI in ALS, and this was detected using the biotin switch assay. And one thing, of course, that springs to mind when we first consider these types of experiments, that these are all involving protein overexpression. So is there any evidence that PDI might be physiologically relevant to ALS? Well, we looked at this in a number of ways, but here's just one study that we did when we looked at the inclusions formed by FUS in ALS patients, and we looked in both familial ALS and sporadic ALS. And what we found was that these inclusions that are the pathological hallmark of ALS seem to be co-localized with PDI. So again, this suggests that there's a strong link between protein misfolding and PDI. There's also now a long list of evidence linking PDI to neurodegenerative diseases. It's been implicated in Alzheimer's disease, in Parkinson's disease. It's known to be upregulated in ALS. But what was interesting was that studies done about 6 years ago identified mutations in PDI and ERP57 in ALS. So it's thought that they're not so much a causative gene in ALS, but they are a risk factor. And I'll mention more about two of these mutations a bit later. So D292N and R300H. PDI and ERP57 have also been shown to have specific functions in neurons. They seem to be involved in the neuromuscular junction and neuromuscular connectivity. So, following on from this study where we looked at PDI in cell culture, we then moved on to look in vivo. And we used here a zebrafish model of ALS. So what we do is we inject mutant SOD1 mRNA into zebrafish embryos. And what we find is that this produces quite a nice ALS relevant phenotype. So we detect these abnormal motor axons, as you can see here in this image, that the axons are short and they're also more branched in the mutant SOD1 embryos compared to controls. We also notice that there's a motor impairment. So when we track their swimming, we notice that they cannot swim as far as the wild type and other controls. So we first examined whether PDI was protective against this motor function, and to use this we use a zebra box system which automatically tracks the distance swum. So you can see this on the image here. So these are automated tracking images. Red records fast movement, green records slow movement, and black represents inactivity. So firstly, you can see in the embryos expressing mutant SOD1 that there is much less movement and there's much more slow movement compared to the controls expressing either wild-type SOD1 or the non-injected controls. However, if we express PDI, then we can restore this movement, and when we quantify this, you can see that we can almost restore the movement of these zebrafish to the levels approaching the wild-type SOD1 embryos. So clearly treatment with PDI restores movement of zebrafish. We then looked at this axonopathy phenotype. So you can see here that on the bottom images, these are the axons of zebrafish expressing mutant SOD1. They're shortened and they're branched compared to the normal controls. And when we overexpress PDI, however, as you can see in the quantification, these axons become the same length as in the controls. So treatment with PDI seems to prevent this mutant SOD1 induced axonopathy. So we then asked, what's the mechanism? Is it this oxidoreductase or this redox activity of PDI? Or is it the chaperone activity which is protective in ALS? So we looked at this in several different ways. Firstly, we used a small molecule mimic of the PDI active site. So BMC had been previously described in the literature. It's a sort of synthetic dithiol structure, and it has a redox potential similar to physiological levels. Its pKa, the pKa of the first thiol also is similar to PDI. We then made some mutants of PDI where we replaced these redox active cysteines, all 4 of them, with serine. So we call this the PDI quad mutant. We then looked at these 2 of these PDI ALS associated mutations, which I mentioned before, and we also looked at the different family members. So many of the family members have the same active site, so we hypothesized that if this active site was responsible for its protective activity, it would be found, it would be common to the different family members. However, to do this, we first wanted to confirm that PDI was in fact modulating a redox environment in our systems, in our cell culture models. So this is where we began to collaborate with Marcelo's team, and he's developed this really interesting biosensor, Clover Ruby. It has these 2 redox active cysteines which are specific for glutathione disulfide. And this is particularly suitable for the time frame of transient expression experiments, which is as we've used in this study. This biosensor also has the Clover, which is used as an internal control for protein expression. So the redox signal can be normalized to the expression level of this reporter. So what Marcelo's team did is express in the cytosol PDI and these 2 mutants plus the quad mutant, and analyze the expression of this reporter using flow cytometry. And the results are plotted as a percentage of the biosensor reduction. And what we see in PDI expressing cells, this is all done with a DMSO as a solvent. And what we find is that there's increased oxidation of the biosensor. It's about 25% of the reduced biosensor by itself. So this again confirms that PDI has redox activity, but it also suggests that PDI is oxidizing in this system. However, in contrast, the mutants did not display this activity. So firstly, the quad mutant, just shown in white, and also the 2 PDI mutants, the ALS-associated mutants. So these mutations are actually present in the B prime domains, and the redox capability of these mutants hadn't really been examined before. So this suggests then that this oxidation that we see in PDI expressing cells is absent from all three mutant cells. So Marcello then went on to look at cells treated with BMC, which, as I mentioned, is a small molecule mimic of the redox activity of PDI. And what we found is that BMC did indeed also induce this intracellular oxidation as compared to the biosensor only. And when we treat cells with PDI as well, we can see that we can even further reduce this oxidizing activity. However, when we look at the PDI mutants, in two cases, when we've got the quad mutant and also the D292N mutant, we find that the BMC actually had no effect on the redox activity. So this suggests actually that these mutants are somehow able to inhibit the activity of BMC. We did see some reduction with the remaining mutant. We also treated cells with BSO. So this is a inhibitor of glutathione synthesis. So this would also inhibit the activity of PDI. And what we found was that this treatment with BSO induced oxidation as expected compared to the biosensor alone, but PDI was much less oxidizing. So it was about 45% of cells compared to 25% of cells with their biosensor reduction. So basically then, all the mutants seem to be similar to cells not expressing PDI, which is implying that they're redox silent. I just want to explain the system that we used to examine the protective activity of PDI against these three mutant proteins, SOD1, TDP-43 and FUS. Now we use cell-based assays overexpressing these different mutants compared to controls, and we used neuronal cell lines and also validated some of these results in primary neurons. Now there's a whole range of different assays that we use, and I haven't got time to explain them all, but I've just summarized them all here. And these represent some of the classic phenotypes or cellular phenotypes associated with ALS. Now here's some of the data we obtained in studies looking at PDI against mutant SOD1, and here we used A4V, which is the most common SOD1 mutation in North America. So we overexpress cells with either mutant SOD1 or mutant SOD1 plus PDI, or that's wild type, or the quad mutant, or we treated cells with BMC. And first we looked at inclusions, and we examined this by microscopy. You can see here in the cells these arrows. This is how we detect inclusions, which is fairly obvious, these compact structures by microscopy. And what we found was that when cells are overexpressing PDI, we can significantly reduce the number of inclusions formed by mutant SOD1. However, when we overexpress the quad mutant, we find no difference to the untreated SOD1. We also examined BMC, and we found that BMC can also inhibit the formation of, formation of inclusions. Here's another phenotype that we looked at, which is ER stress, shown on the right. So here we look at the nuclear immunoreactivity to CHOP, which is a transcription factor that moves into the nucleus when in the presence of ER stress. And what we found again was that in the presence of PDI, we can significantly reduce the activation of ER stress. However, the quad mutant is not protective. However, when we treat with BMC, we do see this protective effect. So this really shows that it's this redox activity of PDI that's protective against mutant SOD1. We then went to look at the two ALS-associated mutations, and we performed a similar type of assay. So I've shown you here inclusion formation. Obviously, protein misfolding is very important in ALS, and we also looked at apoptosis. And what we found is that when we overexpress PDI as we expect, we can significantly reduce both inclusions and apoptosis. However, both of these mutants had no effect. So these appear to be completely inactive and lose this protective activity, which correlates with this lack of redox activity. So PDI is protective against mutant SOD1. Was it also protective against the other proteins linked to us, particularly TDP-43 and FUS? And here's some more data here. This is also from the iScience paper. And when we looked at the protective activity of PDI against mutant TDP-43, and again we used some common TDP-43 mutants. And what we did was overexpress PDI, the wild type, quad mutant and BMC, and what we found was similar to mutant SOD1, that PDI could significantly inhibit the formation of inclusions and also significantly inhibit apoptosis. We've looked at apoptosis by caspase 3 activation here using a specific antibody. However, the PDI quad mutant had no effect on inclusion formation, but what was actually interesting was that quad mutant did show some protective activity against apoptosis. We then looked at the ALS-associated mutants, and here's a different phenotype that we looked at, mislocalization of TDP-43 from the nucleus to the cytoplasm. And what we found was that wild-type PDI could rescue this phenotype. However, the two ALS-associated mutants were unable to do this. So neither of them possess this activity. We also looked at apoptosis, and in contrast to the PDI quad mutant, what we found was that neither of these mutants could actually protect against apoptosis. So again, this suggests that there's something quite inhibitory about these two mutants, that they really have quite harmful effects on the cell. So to further confirm that the redox activity of PDI is protective in ALS, what we did then is treat cells with BSO. So this inhibits glutathione synthesis and therefore inhibits PDI's redox activity. And what we found was that in the presence of BSO, PDI was no longer protective against either mutant SOD or mutant TDP-43. So further adding more weight to the story that the redox activity of PDI is protective in ALS. So that's TDP-43 and SOD. What about mutant FUS? And the short answer was yes, we found again that PDI was protective. And so here's just two pieces of data to show you. This was firstly looking at inclusion formation in cell lines and then validation in primary neurons. So when we overexpress PDI in cells expressing one of the common ALS-associated mutants, we find that we can rescue or significantly inhibit the formation of inclusions. Here's another phenotype that we looked at, and this is quite important in ALS. So inhibition of nuclear cytoplasmic trafficking is now increasingly implicated as an important phenotype associated with ALS, and it's induced by both mutant FUS and mutant TDP-43. And what we found is that we overexpress wild-type PDI, we can inhibit this effect. So this is another phenotype that PDI is protective against. But what we found, interestingly, with mutant FUS is that the results were a little bit different. So we also looked at cells expressing BMC, cells treated with BMC, and we also looked at cells in which the quad mutant was overexpressed. And in contrast to mutant SOD and mutant TDP-43, what we found was that firstly the BMC was protective, again confirming that the redox activity of PDI is protective against in ALS. But we found that this quad mutant was also protective. So this suggests that the chaperone activity, as well as the redox activity, is protective against mutant FUS. And as I mentioned at the start of the talk, There's no disulfide bonds that have been implicated as forming these abnormal aggregates in associated with mutant FUS or wild-type FUS in ALS. So that's one possible explanation for this result. So we've established here that PDI is protective against all three proteins, mutant SOD, mutant TDP-43 and mutant FUS. And this is all in cell culture. What about in vivo? Is the redox activity of PDI protective in vivo? And so we went back to our zebrafish assay, and here's the swimming assay where we use ZebraBox again to track the movement of the zebrafish. And we examined fish treated with BMC and also fish in which the quad mutant was overexpressed in comparison to the wild type PDI. And what we found is that in fish treated with BMC that we do see a protective effect, so we can restore movement in these zebrafish. However, in cells that were in which PDI quad mutant was overexpressed, we lose this protective activity. So, similar to the results found in cell lines, the redox activity of PDI is protective against this motor impairment in vivo. So then we went on further to examine this, and we've looked at different PDI family members. So the common sort of motif is this CXXC active site region, and some of these PDI family members have slightly different intervening residues between the two redox active cysteines, but what we did was look at the ones that have this common CGHC, including ERP72, which contains an extra active site, this A0 domain. And we can look to see whether these PDI family members were protective. So firstly, we looked at ERP57, which is the closest homologue to PDI. And we looked, and this is a paper that was published a few years ago now, but we looked in the cell culture assays that I've shown a few of them today. And what we found was that ERP57 was protective against pretty much all the phenotypes that we looked at. So ERP57 is definitely protective. However, this is not common across all the PDI family members. So ERP72 and PDI-A2, neither of these were protective against mutant SOD1. However, ERP72 was protective against mutant TDP-43 and mutant FUS. So the story isn't as simple as it's just the redox activity of PDI, and this is something that we're looking at in more detail at the moment. And this really just gives a snapshot of a whole lot of different work that was done by another PhD student in the lab, Emma Perry. And what she did was make a whole series of PDI mutations in which we've changed these intervening residues, the CXXC, and examined their protective activity in all of the cellular assays that I've described today. She also made some mutations in the cis-proline loop region of PDI, which is another region which, in terms of the 3D structure of PDI, which appears to be involved in the active site. There's also another couple of approaches that we took, which was looking at the Dsbs. So Dsbs are the bacterial equivalent of PDIs. And PDI contains, as I said earlier, it's an oxidase, a reductase, and isomerase all in one package. But the Dsbs are a little bit different, and that each one has a different one of these activities. So Dsba is more oxidizing, and Dsbc, for example, is isomerazing. So they sort of have these specific activities that are all contained within PDI. And what we found is that we express these normally bacterial proteins, we express them in mammalian cells and looked in our various assays, and we actually found that Dsba was the most protective. So this is the oxidizing form of the Dsbs. So this really suggests that there's a lot of complexity into this this story, and we're working further with Marcelo's team. So he's looking at the redox potential of all these different PDI-like proteins and seeing if we can make sense of this in differing redox activity. So we don't have any conclusive answer. We're still analysing the data, but hopefully we'll get an answer soon and we'll be able to make some firm conclusions from this. And then this is the last few slides show the last pieces of data. So now I want to talk about the location of PDI. Where is PDI found in the cell in ALS? Because the proteins that I've talked about, mutant, SOD, TDP and FUS, they're not conventionally associated with the ER. So that suggests it's the cytoplasm. As I mentioned, TDP and FUS mislocalized to the cytoplasm in ALS. So this really suggests that this is the location where these events are occurring, but PDI is thought of as being in the ER. So we hypothesized that PDI could be working in the cytoplasm in ALS. And so what we did is make a cytoplasmic version of PDI, where we removed the KDL ER retention signal as well as the signal peptide. And then we firstly confirmed, as shown in this slide, that it was actually found in the cytoplasm and not the ER. And then we looked to see whether this was protective in our cellular models. And this summarizes a number of different assays. As you can see, the cytoplasmic version of PDI was indeed protective in cells expressing mutant SOD1 and mutant TDP-43. So this suggests that, yes, it's the cytoplasm. This is the location where PDI is displaying protective activity in ALS. But again, the situation is a little bit more complicated in that we've also examined another phenotype that's been linked to ALS. So DNA damage is now increasingly implicated, and impairment of DNA repair processes seems to be an important process. It's been recently shown that TDP-43 and FUS are both involved in DNA repair, and it appears to be a loss of function that is implicated, although toxic gain of function is also implicated generally in ALS as well. And so we hypothesized that maybe PDI is protective via its redox activity, and redox dysregulation has also been implicated in DNA damage. So we wondered if PDI might be protective against DNA damage, including that induced by mutant TDP-43. And this is a paper that's currently on bioRxiv. And what we did was examine this in cellular models again, and also in zebrafish. And what we found was that PDI was indeed protective against this induction of DNA damage, including in cells expressing mutant TDP-43. And what was also interesting was that, as I've just mentioned, PDI moves into the cytoplasm in ALS, or appears to be functional in the cytoplasm. And there's actually been a study from a couple of years ago that's shown that there's a protein reflux system that facilitates the movement of PDI into the cytoplasm when in the presence of ER stress. We look to see where PDI might be active against DNA damage. I mean, DNA damage occurs in the nucleus. So we looked to see whether PDI was indeed located in the nucleus in ALS. And what we found was that in response to DNA damage, PDI does indeed move into the nucleus. So this suggests really that PDI is able to move throughout several locations within the cell when it's needed in response to stress. So, in summary, I've shown you today that our work's shown that PDI is broadly protective in vitro against mutant SOD, mutant TDP-43 and mutant FUS in ALS. ERP57, its closest homologue, is also protective in vitro in these cellular models. However, PDI is able to protect against important behavioural phenotypes associated with ALS. So it restores this movement impairment, and it prevents this axonopathy in zebrafish models of ALS. PDI appears to be protective in the cytoplasm and the nucleus, and really it's this oxidoreductase activity of PDI that seems to be mediating this, this activity. We have seen in some instances that the chaperone activity is also protective, but by far the most common and the most important property is this redox activity. So we're doing further work, and I haven't got time to describe any of this today, but we're looking in animal models of ALS, in mouse models of ALS, to see whether PDI is a novel therapeutic target. So in the normal situation, PDI has an important role in the formation and isomerization of disulfide bonds, but in disease states, it appears that PDI also has a very important role, and that possibly both loss of function and gain of function are important. So loss of function leads to protein misfolding and all of these phenotypes that I've talked about today, but also potentially this gain of function by this S-nitrosylation that we detected in human patients. So thank you for attention. I'd be happy to take any questions. Thank you.